First question you need to answer is, why do we age in the first place? I mean, what really is aging? I've made a video in the past about telomeres. These are the end caps on your chromosomes, and every time a cell divides, the telomere gets a bit shorter. So it was thought that these telomeres are kind of like the tips of your shoelaces, and they prevent the chromosome from fraying. But there are other signs in older bodies that you have old cells. There are an accumulation of things that are called senescent cells. They're essentially these zombie-like cells that just go on living in your body and inflaming the cells around them. There's poor intercellular communication. There's mitochondrial dysfunction. Those are the powerhouses of the cell. There are these 8 or 9 different features of older cells, and they are the hallmarks of aging. But the question is, are they the cause of aging, or are they kind of the result of a deeper root cause? In the middle of the last century, the hypothesis was that it was damage to our DNA, mutations to our DNA that happened over the course of our lives that led us to be older. But evidence since then has suggested that that is not really the case. I mean, you can take an adult cell and you can clone it into a new organism. And that organism appears to live about as long as non-cloned organisms of the same species.
Now, the first sheep, Dolly the sheep, had a short lifespan. She died early. But cloned animals, you can now clone a monkey, you can clone dogs. In fact, Barbara Streisand, the actress, she cloned her dogs. And they're expected to live a normal lifespan.
So in that way, it seems like all the information is still there in the DNA. So if we're not losing information in our DNA, then what is the reason for aging? Well, Professor Sinclair suspects that it's a loss of information, but not the information in our DNA, in our genome. No, Professor Sinclair suspects that the loss of information is in our epigenome. So what is the epigenome? Every cell in your body has the same DNA, but different cell types have different epigenomes. They have different ways of packaging that DNA, coiling up, a lot of it so that it's not red, and leaving some parts of the DNA spooled out so it's easier to transcribe and turn into proteins and run that cell. So the epigenome is responsible for turning on or turning off different parts of the DNA. And the way it does that is with proteins called histones that essentially the DNA is wrapped on, and also things like methylation. So there's these chemical signaling markers which are placed on the DNA in certain positions. So the idea is when your body is first forming, the epigenome is what tells your cells what type of cell to be. But as you get older, Professor Sinclair's hypothesis is that we are losing information in the epigenome.
And that's important because if a skin cell needs to remain a skin cell, that's the epigenome. And if you don't have the epigenome, the skin cell will forget what type of cell it is, and it might turn into a brain cell, which may not be that bad, but if your brain turns into a skin cell, you've got a problem. And I think that's largely what aging is.
I've got to say, there's some weird, hair patches, on my shoulder that have happened as I've gotten older. Is that a cell, doing the wrong thing? Are those meant to be skin cells? Are they screwing up? Or is this just some, I don't know.
No, weird stuff happens when you get older, right? You start to get hair growing where it shouldn't, ears, nose, back. That's cells losing their identity. Cells go, I can't remember what I'm supposed to do. I'm not reading the right genes anymore.
So the key to this sort of breakdown of the epigenome is DNA damage?
Yeah, so when you go out in the sun, and not like today, but on a day where there's a lot of sun, you'll break your chromosomes. And in the effort that the cells go to stick the chromosome back together, the DNA isn't just flailing around, it's actually bundled up. The cell has to unwrap it, recruit proteins to help, join it together, and then they have to go back and reset the structures. And that resetting of the epigenome happens about 99%. That 1% is the aging process.
So over time, histones are not returned to the right places, and DNA methylation is added in places where it shouldn't be.
We can read that methylation pattern, and I could tell you how old you are, exactly, and when you're even going to die.
How could you tell that?
Well, it's a clock. We call it the Horvath clock, named after my good friend Steve Horvath. And so these little chemicals that accumulate on the DNA, like plaque on the teeth, we can read that. And the more you have, the older you are biologically. So you might only be 40. You're younger than 40, of course, but I'm 50 now, but I might be biologically 60. Actually, I was, and I changed my life. And then the test said I was biologically 31.
I mean, one of the things I found really interesting was you found a way to make mice age faster. So how'd you do that?
Well, the clock of aging is due to the loss of the information in the cell. And one way to accelerate that is to go break a chromosome. Instead of going in the sun, we engineered a mouse where we could break its chromosomes. Not enough to cause mutations. The cells put the DNA back together, so we didn't lose any genetic information. But if we're right about the epigenetic information theory of aging, those mice should get old. And that's exactly what happened. It's gray, it's got a hunchback.